Hello everyone, and a warm welcome back to our Evolution and Ecology seminar series. My name is Elizabeth Duxbury, and I'm a postdoc in the research group of Alexei Maklikov at the University of East Anglia, and I'll be hosting today's session. This week marks the one-year anniversary of launching our series, and myself and fellow co-organisers, Andreas, Julia and Wouter, have been honoured to host such a diversity of great minds so far, and we are grateful for the continued enthusiasm from the research community for this initiative. So thank you all for your fantastic engagement in the series. As always, there will be a Q&A session directly after this talk, so please post your questions in the dedicated Slack channel. And with that, it is my great pleasure to introduce today's speaker, Professor Tracey Chapman, Professor of Evolutionary Genetics in the School of Biological Sciences at the University of East Anglia here in the UK. Tracey's research explores how interactions between the sexes drive evolutionary change by probing the mechanisms underlying sexual selection and sexual conflict and their ultimate consequences for fitness. During a remarkable career, Tracey's research has elegantly revealed how seminal fluid proteins, and particularly sex peptides, that is transferred from males to females during mating, are instrumental in mediating the spectacularly diverse range of physiological, behavioural, and transcriptomic responses of female Drosophila melanogaster fruit flies to mating. More recently, Tracey's work has focused on the social and sexual responses of both females and males to sperm competition, and the strategies used by females to resist male-induced fitness costs. In addition, Tracey is actively involved in projects exploring the intriguing role of the gut microbiome in mate choice and the role of nutrition in shaping sex differences in lifespan, the latter of which I had the pleasure to work on for my PhD with Tracey. Tracey's research is continuing to make important contributions to our understanding of how reproductive traits evolve and the genetic basis of sexual conflict in Drosophila, whilst also translating this fundamental research into developing novel genetic methods for controlling insect pests. Tracey has published numerous high-impact papers, was the holder of the prestigious Royal Society University Research Fellowship, and is the current editor-in-chief of the journal Evolution, as well as being a strong advocate and driver for equality and diversity initiatives. Today, Tracey will present her talk entitled Sexual Selection, Sexual Conflict and Evolutionary Change. So Tracey, thank you for accepting our invitation, and over to you.
Thank you very much. Okay, thank you very much, Elizabeth, for that really warm introduction. I really appreciate it. And I'd like to thank the organisers hugely for the opportunity to present in this wonderful symposium series. And I do want to thank the organisers for organising all of this whole fantastic series, because it's been a real beacon of light, I would say, over the last year, which, let's face it, has been actually sort of rather awful, appalling year for many people. So, thank you. I think this has been a great endeavour, and I'm really pleased to be invited to have the opportunity to present. Okay. So when we think about sexual selection, we're considering elaborated traits in males that serve to facilitate success in competitions, same-sex competitions for access to females and to fertilizations. Or we're considering elaborated plumages and colours that serve as the basis for female choice. But sexual selection, so that is the selection that's focused on ensuring success in reproduction, is not just about the evolution of exaggerated traits. It has genome-wide significance because measures of quality capture variation across many, many different loci in the genome. And so success of high-quality individuals or choice of high-quality individuals by females has a genomic imprint. And theory and experimentation shows us that these effects can purge deleterious mutations, can protect against extinctions, and can increase invasiveness. But I think in this sort of beauty contest of sexually selected traits. I think the invertebrates always win hands down, and so or probably legs down it should be. But here are some of my favorites, and so some elaborated jaws. The stalk-eyed flies, where the eyes on the ends of the stalks are quite literally used to eye up other males, but also serve as the basis for female choice. The extraordinary peacock spider, which just defies any kind of explanation. And also in the bottom right, this is a photograph which serves to remind us that sexual selection is very important both before and after mating. And these are adaptations in the seed beetle which serve in sperm competition. And I think, you know, the balance of pre and post mating sexual selection is itself incredibly interesting. But we've known since the 1970s and the seminal work by Jeff Parker that interactions between the sexes can be shaped by conflict as well as coordination. And this stemmed from observations on creatures such as these, and this is yellow dung flies. And these observations showed that the competition between males can be so strong that the females can get damaged and even drowned in the dung when they come to lay their eggs in these cowpats. And so it was realized that you could get very, very strong selection on males, even despite the cost that it causes in females. And so elaborating on that theme somewhat, so we're thinking about reproductive traits whose value emerges as an emergent property of interactions between the sexes. So, for example, how much to invest in any particular reproductive event or how often to mate. But the value of that trait, the optimum value of that trait, is different for males versus females, implying a zone of conflict signified by the grey arrow here. And because a trait like this can only take one value, both of those optima can't simultaneously be realized. You know, there's only one particular amount of investment that can be made in any particular reproductive event. So those optima can't be simultaneously maximized, implying a potential for conflict, because females would prefer or gain fitness if the value of that trait was slightly lower than it is, and males slightly higher than it is, or at least in the way that I've drawn it here. And drawing it out this way, I think helps us to think about some properties or some features of this system. So how far apart are these optima? And these are going to govern, you know, how intensely conflictual these interactions might be. What is the current value of the particular trait? What's the relative power of each sex to affect what the value of this trait should be? And is there even any mechanism which either sex can use to change the value of such a trait? Is there any genetic variation for selection to act upon? And if you like, this is the kind of motive, means, and opportunity of the system. So this is a true evolutionary conflict between different genes in males and females. Now, of course, it's important to emphasize that, of course, the sexes have to coordinate to reproduce successfully, but we've got small and large gamete producers, which are generally males and females. We've got costs and benefits varying of different making those gametes and the consequences of having different sized gametes, outbreeding, non-monogamy, and those sorts of features, which are very, very common in mating systems, will mean that fitness interests of individuals that come together to reproduce are not always aligned, or maybe not even aligned very often. And this has the potential to drive evolutionary change. And this has been realized by many researchers, and some of their work is indicated at the bottom here. So, of course, the wider significance of these sexual interactions, which are driven by sexual selection and influenced by sexual conflict, is that they fuel evolutionary change. So this is fundamental importance. But I think it's also important to think about the significance in terms of perhaps things like state-of-the-art insect control strategies. And this is the Mediterranean fruit fly. So this is a global agricultural pest that we also work on, but I don't have time to talk about that today. But current strategies being developed either gene drives or that employ under dominance or various kind of toxin antidote systems, generally rely on the release of insects into the pest population, and rely on the requirement that those released individuals are somewhat competitive, and actually that some of them mate with the target population to effect control, whatever that control is. And so obviously the sexual interactions between these released individuals and the wild individuals is of crucial importance in determining things such as the threshold needed, how many insects need to be released, how successful they are, how often they need to be released, and in order to get any kind of control. So this is another wider significance of the need to understand the full range and the full complexity and richness of sexual interactions. Well, that's the end of my introduction. So I want to talk about two things today. So the potential for conflict first, and why the evidence that shows us that interactions between males and females can be shaped by conflict, as well as all of the coordination and sexual selection. And then there's a difference between having a potential for conflict, which might be incredibly widespread or might even be universal, and that conflict actually being expressed. And so what are some of the key determinants? And we heard about some important determinants, in fact, in the excellent talk by Anil Agrawal last week. He was talking about the complexity of the mating environment, but I'm going to focus here on two key factors: resources and the social environment. Okay, so first, the potential for conflict. And so we've got small gamete producers, we've got large gamete producers, we've got differences in the consequences of reproduction for each sex, and each sex, maximizing their fitness in different ways. A picture here, because it took me a long time to make this in PowerPoint, so partly I just want to share this with you because I was quite proud of having made it. But it also serves to remind me to say that I'm going to talk about our work with the fruit fly model system, Drosophila melanogaster, because of the tractability and all of the genetic tools that we can use. But we are talking about fundamental concepts in interactions between the sexes. And so the significance of the things that I'm talking about, I think, does transcend this particular experimental system. Okay, so anyway, I want to start by showing some new data on a phenomenon that's been known about for decades, which is the significant differences in the consequences of reproduction for each sex. So that's shown on this slide here, and I'm going to walk us through it because there's quite a lot on here. And so this is a slide to show for females, which are on the graph at the moment, and males, which I'll put on in a moment, how the consequences of both reproductive activity and also same-sex interactions differ significantly for females and for males. And those consequences are described here in terms of survivorship. And so the initial mortality rate on the x-axis, so aging starts earlier as the x-axis increases, and on the y-axis, you have the rate of aging increases. So the actual rate at which aging increases once it starts, and that's on the y-axis. And we've got dots showing the response of different treatments, 4 different treatments in females shown. And so these are in grey, individuals that reproduce once at the start of their lives, and then are just subject to same-sex interactions for the rest of their lives. We've got intermittent reproducers in pink. We've got continuous reproducers in red. And we've got individuals in black in a density control. So these as well are individuals that reproduce once, but at double the density. And so they have a much higher capacity for same-sex interactions. And each one of these points is 5 replicate cages. So we keep them in the boxes which are shown in the bottom the bottom right-hand corner. So of individuals of 2 to 400 in each box. So we've got 5 boxes. So this is a large-scale experiment, but the individuals are interacting freely in this kind of complex, big box environment. Actually, what you can see is that for females, the consequences of increasing reproduction shown by the solid black arrow and increasing same-sex interactions is very different. So the former increases both the initial mortality rate and the rate of aging, so speeds up the rate of aging once it starts in females that reproduce at ever higher rates. But increased same-sex interactions does something different. Has an effect on initial mortality for sure, but even seems to decrease the rate of aging. But the real reason for showing you this diagram is that I wanted to contrast the pattern in females, which I've shown here in the circles, with what happens for males. There are 2 important things here. One is that the magnitude of these effects is very much smaller, so they're very much diminished in their effect size, but also that the pattern of what happens when you increase reproduction versus increase same-sex interactions is completely flipped. So it's completely different in males as opposed to females. So this is just indicating, I think, in perhaps reasonably rich way, really how different the consequences for reproductive events are for each sex. Okay, so back to the potential for conflict. Well, these different consequences of reproductive events in terms of the benefits and particularly the costs, especially those large ones in females, perhaps suggest that the expression of the effects of adaptations in one sex can draw the other up their optimum. And so this is the central sort of thesis of sexual conflict. And when we were searching for the explanation for costs, and particularly these very large costs that we were seeing in females, we became very interested in the effects of this extraordinary group of molecules, the seminal fluid proteins that are transferred along with sperm by males when they mate. And they are largely made in these large paired accessory glands. And these are these beautiful structures here shown in the photographs on the right, with these 2 types of secretory cells. So we were interested in these molecules because they are structurally diverse. They range from small, simple peptides, pro-hormone-like molecules are very big things, and this is all work from Eric Kubley's lab and Mariana Wolfner's lab. But the really interesting thing is the extraordinary range of fitness-related effects that these peptides have on females when they're transferred from males during mating. So this slide summarizes an absolutely enormous amount of research from different laboratories. And I refer you to the wonderful talk by Mariana Wolfner in this series where she talked about some of these things, talked about some of these phenotypes. And so there's a range of different fitness-related effects here that these the transfer of these individual peptides collectively as a group, and there are about 200, more than 200 in total that we know about. And that's the latest estimate from Stuart Wigby's group. But there are some which I think we can think about, particularly in terms of the phenomena that we've been discussing so far, that are perhaps potential candidates for mediating conflicts of interest between the sexes and altering the balance. So in terms of investment in reproduction, there are seminal fluid proteins that affect how many eggs females ovulate and how many they lay, how much they eat, what they eat in order to support that higher rate of reproduction, but also seminal fluid proteins that affect the receptivity, the attractiveness as females. So are going to impact directly on the remating frequency and the extent to which females are subject to the receipt of courtships from males. And so this is what sort of led us to investigate the possibility that seminal fluid proteins had something to do with mediating conflicts of interest between the sexes. And the evidence actually that effects of seminal fluid proteins can draw females away from their optimum comes from a number of different experiments, but I'm just going to describe 2 of the ones that we've done here. So if we reduce seminal fluid protein transfer by males, we can eliminate costs of mating in females. So we've known about that for a long time. And we can also demonstrate that the transfer of seminal fluid proteins can benefit males and cause costs in females. Now, that's not to say that these effects are always expressed, because what we're trying to develop here is a kind of richer understanding of when these effects are most felt, when they're most important, what are the factors that determine them. But I think these are quite strong pieces of evidence that show that these interactions can be shaped by conflict which arises through the actions of these seminal fluid proteins. And so perhaps actually giving an example from a more recent piece of work that we've done. So here's some evidence that the effects of seminal fluid proteins can actually influence the balance of interests between males and females. So what we've got here is a triangle setup. So a realistic setup. Most wild-caught females have the sperm of at least 2 males. And so this is a triangle where there are 2 males that mate in series with a female. So male 1 and then male 2 mate with a female. And what we can do is we can experimentally lengthen or shorten the interval between the mating of male 1 and male 2 and ask, what is the interval which maximizes the offspring production for each party in this triangle? So what is the remating interval that maximizes the number of offspring that the first male fathers, that the second male fathers, and what is the interval which just maximizes the total number of offspring that the female produces? And so if you do that, actually what you find is the number, so the optimal remating interval is different for each of the parties in this triangle. For example, the first male gains, not surprisingly, by having quite a long remating interval between his mating and when the female mates again, because he can get more offspring in the meantime. And not to go into why these numbers are exactly what they are, but the point is they're all different, okay, which reflects, I think, the potential conflicts of interest in the system. And interestingly, what happens if you remove the ability of females to respond to a key seminal fluid protein called sex peptide, the optimal remating interval, so that interval which maximizes the number of offspring produced by each individual in this triangle, converges, becomes very similar. So that's interesting, because if you silence this pathway, you take the seminal fluid protein responses out of the system, the interests of the parties involved converge. And there's a lot of raw material here too. So in this slide and the next slide, I'm going to talk about the amount of genetic variation that there is and show that both the amount of seminal fluids that males actually put into females varies. There's genetic variation for that trait, but also the response of females to seminal fluid proteins shows genetic variation. So what's shown on this slide is the relative concentration of a seminal fluid protein, sex peptide, that's actually transferred into females by males from these Drosophila genome resource panel lines. And so this is the set of isolines developed by Trudy McKay's group. And so there is restricted genetic variation within these lines. So you can take significant differences between these lines with respect to the trait of interest as evidence of genetic variation in that trait. And so these are males from these iso lines mating with standard females, and we are using a immuno-quantitative PCR to pick up the actual amount of peptide that males are transferring into females. And the point, there's a no template control over on the left-hand side, but there's significant variation here. So there is significant genetic variation in the amount of this seminal fluid protein that males put into females. There is also genetic variation, and this is for the same seminal fluid protein, in how responsive females are to it. And so here we're taking females from these Drosophila genome reference panel lines and mating them with males that deliver a standard dose of sex peptide, of the seminal fluid protein. And in this case, what we're looking for is a difference between the open bar and the filled in bar for each of these lines. So have we got evidence of females that are incredibly responsive? And here the trait of interest is the extent to which the fecundity of the females is boosted by receipt of this seminal fluid protein. And just to illustrate, we do indeed have evidence for genetic variation. So we've got some females that are very responsive, whose fecundity is boosted incredibly significantly by receipt of this seminal fluid protein, and others where we don't. So there is a lot of raw material for selection to act upon here. Interestingly, the rate of signal and sort of receptor coevolution doesn't necessarily run equivalently across different species, but I don't have time to talk about that today. Okay, so returning back to the theme, well, if seminal fluid protein effects can indeed draw females off their optimum, what we should predict is that the evidence of costs, and so females being dragged under these experimental settings off their optimum, should select for counter responses. And so is there any evidence that that can happen? And we can ask that question by using an experimental evolution paradigm. And so we create different sex ratio, adult sex ratio environments, either female biased, equal sex, or male biased. And we have replicates of each and a census size of 100 in the 3 replicates of each of these regimes, and we allow them to run over many generations. And then what we would predict, and in fact what we see, is that we see an elevated, very much higher level of courtships per female and numbers of matings per female in the male bias regime. And so the question we're asking here is, well, how do females respond to those differences in sexual selection and sexual conflict? And we can do that by taking females from those experimental evolution regimes and mating them to standard males and asking, have the costs of mating, so the costs of being exposed to males throughout their lifetimes, have they been ameliorated in the way that we predict? And that is indeed what we see. So we look at the survival of females continually exposed to males. It falls out in the way that we predict. Survival is higher in females drawn from the male bias regimes than it is from the equal sex than it is from female biased. So this is evidence that suggests evolved resistance in female responses. And we have done this experimental evolution experiment completely independently twice. So this is, and got the same result. So we think this is a robust finding. Okay, so I've talked about the potential for conflict, key determinants. So let's look at some of the factors that might determine how often this potential for conflict is realized, and that will help us develop a richer understanding of the importance of these processes, how often conflicts are expressed, whether they're important now, whether they've ever been important in the past, and this kind of thing. Okay, so resources are important because, of course, they limit the capacity of an individual, or can do, to express its genotype or to respond. And one example of that that's relevant to this context that I've just been talking about are nutrient-dependent female responses. And so the experiment that I was just describing, using these experimental evolution paradigms, was done on a rich adult diet. And so there was the opportunity for individuals to feed ad libitum and potentially to express their genotypes with low constraint, one assumes. But we also did this whole experiment, this whole experimental evolution paradigm, under nutrient limitation, where we restricted the amount of protein, yeast, 20% of the normal amount, standard amount, in all of the same regimes. And so to test for nutrient dependence, for condition dependence in these responses. As far as we can tell, interestingly, the selective environment is very similar in both of these regimes, in that we see equivalent levels of courtship and matings, and we still see more courtships and matings per female in the male bias regimes in comparison to equal sex and female biased. And so as far as we can tell, the selective environment is the same in both nutrient environments here. But if we test again for nutrient-dependent female responses to males, so asking the question, can females from the male bias regimes ameliorate costs of reproduction? Can they evolve resistance to whatever it is that males are doing that is costly to them? Then the answer is shown here. And so the rich diet on the right-hand side, these are the results I just showed you a moment ago. But it doesn't happen on the poor diet. So we don't get that evolution of resistance in those females. So it suggests that female responses to sexual selection, sexual conflict, this elevated level of conflict that the male biased females in particular receive generation after generation is constrained under nutrient limitation. So that's one example of how resources are important. And of course, there are many more. But I wanted to focus in the the last part, really, final part of the talk, in the social and sexual environment. And this is important because it will affect the strength of sexual competition and potentially also the strength of resource competition, perhaps among females for access to overposition sites. So what we're really asking is the intensity of the interactions between males and females and the capacity to be shaped by conflict, as well as all of the things that need to go right. How is that impacted when we introduce and broaden out and add complexity to the social and the sexual environment? So first of all, I want to talk about the responses of males. And after all, we think that, as I laid it out at the beginning, this is a primary determinant of the intensity of interactions, conflictual interactions with females. So we expect and we predict that the sexual environment of the males should have some influence here. So we know that males show very strong plastic responses to other males. And specifically what's shown here is the length of exposure of males to other males prior to mating as a function of mating duration, how that relates to their subsequent mating duration when they do meet a female. And what you see, there's quite a strong relationship here. So the longer that males are exposed to other males prior to meeting a female, the longer they will mate for when they do meet a female. And so in the mating arenas, it's just one male and one female. There's no interference at this point. So that's a really strong effect. And the benefits of those extended matings for males are very striking. So females that have mated to males that were previously exposed to a rival will take longer to remate. Females that mate with males that have previously been exposed to a rival show increased fecundity, and males that have previously been exposed to a rival gain a higher share of paternity overall. So these are very significant fitness benefits for males. So males are responding in some way to the threat of sexual competition by mating for longer, and they gain these fitness benefits. We know something about the transcriptomic basis, so the underlying basis of what's actually going on here. And this is a summary of a gene expression piece of work that we did, which looks at what are the transcriptomic differences, responses in males that are simply exposed to conspecific rivals for 2 hours, 26 hours, 50 hours. And there are lots of things that change. And in the head and the thorax of the males, there's an activation of lots of sensory genes and all these other kinds of genes. In the abdomen, you have as well a really striking early upregulation of immune genes and ejaculate component genes. And that's the thing that I wanted to emphasize here. So males, they perceive other males, and very rapidly, within two hours, we can see this burst of transcription of ejaculate component genes, presumably in response to the threat of sexual competition. And the interesting consequence of that, I think, is shown here. And this was work that was done with Stuart Wigby and Mariana Wolfner. And so this response of males to rivals actually initiates a plastic allocation of more seminal fluid proteins being transferred into females. So what you've got is male equivalents of the amount of sex peptide that's transferred into the female. There are 4 different lines here. It doesn't matter what blue and red means in this context, but what's important is that in each case, there's more of this seminal fluid protein, sex peptide, transferred into females if the male was previously exposed to a rival. So in each case, the squares are higher than the circles. So males are being exposed to rivals, perceiving a threat to competition, turning on the expression of all of these ejaculate component genes and actually putting more of seminal fluid proteins into females. Why this is important in terms of the intensity of the interaction between males and females is that it has the potential to exacerbate conflict. We know that the receipt of these molecules, well, can induce lots of phenotypes, and the effects of these are potentially costly. And subsequent work from Stuart Wigby's lab has shown that there's even modulation of the components, the way that the ejaculate is made can even respond to the social and the sexual environment. We know that males detect rivals in an interesting way via complex sensory cues, and we work this out by eliminating in turn and then in combination these different cues, so sight, smell, touch and hearing. And what we're trying to do here is work out the basis of how do males know who's in their environment. And what we can do is ask which of these cues, when we take them away, make males unable to respond to rivals. And so we're looking at the point at which these significant responses to rivals goes away, are lost. And that's summarized in this table here. So at the top, we've got when we take away single cues, and there are multiple rows for each because we can do this in different ways in Drosophila melanogaster, either genetically or through other manipulations. And so if we take away just single cues that prevent males from hearing each other or smelling each other or seeing each other, that makes no difference to their capacity to respond to rivals. Where it gets more interesting, where they lose the ability to do that, is when we take away combinations of cues, and particularly those that involve these first 3 sensory modalities here. And if I just summarize this finding, the results show that males detect their rivals by using 2 from any 3 of the cues of smell, touch, or hearing, and it's hearing song. So they can detect 2 out of any 3 of those cues, and they respond to rivals. Why they do it in that way, and why it's not just one cue, and why this sort of redundancy in the system is an interesting question. But our thoughts are that this is a reliable way to know who's in the environment. It's robust to behavioral phenotypic noise. And you can also, it offers the potential for males to integrate different components of the environment. Furthermore, male responses to competition also evolve. And we can ask this question again by deploying these experimental evolution regimes. And here we can take males from these regimes, put them with standardized females, and ask whether there is an overall sort of baseline level response to perceived levels, actual levels of competition. And what we see is this result from Alice Dorr. Male biased males evolve longer matings overall. So male biased males that are in continually elevated sexual selection, competing with other males, conspecifics for matings all the time, very high levels. They evolve longer matings overall. And this is a subtle effect, but it's significant, and it happens in both of these nutritional regimes. And so this is something which isn't nutritional, doesn't show nutritional dependency. So that's one thing that happens. Perhaps even more interestingly, the plasticity patterns of males when they come from these experimental evolution regimes also differs, also evolves. And we've shown that in this kind of design. So if you take males from each of the experimental evolution regimes and you keep them prior to mating, either with no rivals or with a rival, and this is a standard type of base stock male. So we keep them in those environments for 3 days, and then we put them with a female and we ask, well, is there any evidence that the plasticity that we see itself has been affected by the sexual conflict and the sexual selection history of these regimes? So they sit with the males and then for 3 days, and then they mate with females, or they get exposed to females. And there's some interesting things that happen. So I'm just going to illustrate with a video. So this is a control. So this is an equal sex regime male. So the male's on the left, female's on the right. This is what happens when, so this is a male that's been exposed to a rival. And he's just demonstrating the sort of standard courtship repertoire, or some of the elements in Drosophila melanogaster. So the male is starting to sing. I don't know if you can see, male on the left, he sticks his wing out to the side. He's clearly orientated towards the female, is trying to sort of, there was a half attempted copulation. He's clearly following her around. He's orientated towards her. He sings again. It's around the front, and so the elements of courtship that are occurring, and he's clearly following and chasing her around. Okay, so that's us. Interestingly, and this is going to be the sort of equivalent of you know, observer watching these, you know, watching paint dry. This is a male bias male, so a male drawn from the male bias regimes, which has been treated in exactly the same way as the individual that I just showed you in the previous video. If I start this up, so the male is on the left, female's on the right. This is a standard female, you know, they're quite active, she goes around, and the male is grooming. Male does nothing. I'll let you watch, nothing much happening for a while. And so it goes on. So something very interesting has happened to the pattern of plasticity expressed by males in these, drawn from these male bias regimes. But it's not that these male bias males don't do anything at all. They retain the courtship repertoire, and that is seen in the conditions when they're not exposed to rivals. But when they are exposed to rivals, they actually switch off most of the courtship repertoire. So it's a change in the plastic behavior of these males. And that's illustrated. So this is the quantification of the courtship videos that I just showed you here. So male bias males, they show reduced levels of orientation, singing, chasing. So the reduced representation is shown by the blue arrows, and also attempted copulations. So the pattern of plasticity itself can evolve in response to these regimes. Okay, so I think that these results collectively show us clearly that responses of males to sexual competition can clearly alter the intensity of sexual interactions with females. What about the responses of females? And so this is some new work that I'm going to report to you, newer work. And so we expect, what happens when we expose females to other females is it has no effect on mating duration, and seems to, the responses that I'm going to show you seem to differ quite markedly in the kind of phenotypes involved and the cues as to what happens in males. So females might respond to other females in terms of resource competition or even sexual competition. And so what we have found is that females that are exposed to other females prior to mating actually lay significantly fewer eggs after mating. So what we see is marked fecundity plasticity responses in females according to their same-sex social environment. So there's a focal individual female just above the A and B, and the pre-mating exposure is 3 days of exposure to either no other individuals or 3 other individuals. And what you see when you then put the females on their own and they lay eggs for 24 hours is that those were previously in a group lay significantly fewer eggs after they mate. But this is not a sex, not just a conspecific effect. And if you expose females in Melanogaster to simulans or Yakuba, so these are 2 other species drawn from the Melanogaster species subgroup, you still get this effect. So it kind of implies that this is a response to something to do with oviposition site usage. I mean, these species do, or competition for food, these species can co-occur, and they all use the same kinds of rotting fruit resources in the natural environment. Okay, so this is the response that we see, and it's very robust. So we started trying to set out again to find out, well, what causes these responses? What's the nature of the cues? What do females use to know who's in their environment and actually to change quite dramatically their fecundity? On the left and on the right is the same experiment. These are just 2 replications of the same piece of work. And this is the result that I just showed you. What we're going to do now is just try and pick apart some of the contributions that females might be picking up on to try and isolate the mechanism involved. So when you expose a female to other females, you're not just exposing them to the smells and the sounds. There are other things. You're exposing them to the eggs of the other females, and that's the little oval with the 2 thingies, and deposits. And I mean, deposits isn't some sort of horrible euphemism, but I mean, deposit means probably it could be insect poop, but it could be you know, things from the proboscis of the female, other elements. And so those are the kind of, you know, major components. So it could be something to do with the females themselves, or it could be to do with the eggs, or it could be to do with the deposits. And so we tried to take away these elements and then see if we still get this change in fecundity. So if we expose focal females for 3 days prior to the event here to females that can't lay eggs, so there's no eggs here of other females, but there are still other females deposits, we still see this drop in per female fecundity. Okay, so it's not eggs per se. If we go just over to the right-hand side, if we have no other non-focal females in the environment, but we just have eggs and we have deposits laid by the females that laid those eggs before they were removed, we still see this effect. So it suggests then that the deposits might be the thing that females are queuing into. And again, if we just expose focal females to the deposits of others, we still see this plasticity in fecundity quite markedly. So that predicts that gustatory, perhaps tactile cues are being used by females. And so again, following the sort of parallel strategy that we did in males, we can eliminate in turn these different cues and ask which of them, when we take them away, make females unable to respond to other females and change their plasticity. So here what we're looking for is the point at which the removal of the cue makes the difference between the blue bars and the red bars go away. So, because we're asking, when do we see the loss of the ability to make this response in fecundity? And so these are 2 different ways of altering the ability of females to smell other individuals in their environment. And these responses are retained. So we don't think it's to do with that. If we do the experiment in the dark, and so we prevent visual cues that way, we still see these responses. So we think that vision is not being used by females, because if they can't see each other, they can still reduce their fecundity in response to the presence of other females. But when we prevent tactile cues, we see that this effect goes away. And in each case, we've got the control on the left and then the cue removal on the right. And so when we prevent tactile cues, we don't see this response, this plasticity goes away. So it suggests that we've identified tactile gustation as a cue used by females. And so this also shows us that, you know, the plastic responses of females to their environment can alter the intensity of sexual interactions by altering investment patterns, you know, how many eggs are laid. But also it shows us that even in females that receive these fecundity enhancing seminal fluid proteins from males, they can tailor their response to that according to what kind of environment they're in. These results collectively also tell us, I think, that the basis of these plastic response phenotypes and the cues used are really different in both sex. And also we have a bioarchive preprint which shows the ways in which plasticity expressed by females and by males actually interacts to determine the expression of some of these traits. So I don't have time to talk about that today. So just in summary then, so I've talked about the potential for conflict, how we think that this will shape this system, how that potential for conflict is affected by resources, the amount of resources in the system, and these really interesting and complex social environment effects, which are very important, very striking, and we are just really starting to build a picture, I think, of their complexity and their interest and how they're important. If there was a take-home message from today, I guess it would be that of course the sexes coordinate to reproduce successfully, but that these interactions can also be subject to conflict, and that this conflict is an important force for driving evolutionary change. And so it just remains for me to thank members of the team, my team, and colleagues that I work with in current work and work that I've talked about today. The bits of work that I've talked about, the people who contributed to the bits of work that has been featured most prominently are marked with an asterisk. And of course, I need to acknowledge the funders who supported this research. So thank you very much for listening, and I'm happy to take questions.
Thank you very much, Tracey. That was a really fascinating and enlightening talk, and I think a real tour de force through a vast majority of your fascinating research over the years. It's fascinating to see how you have described both the potential for conflict and also some of the key determinants in terms of resources and also the impact of the social environment. And I think it's fascinating how you also show the great capacity for plasticity as well in the system. And while we wait for the audience to have some time to type in their questions, I thought I would start off with a question of my own for you. So I'm quite curious. So obviously you've talked a little bit about how females may be able to develop some resistance mechanisms to counteract some of the costs from males via the transfer of the seminal fluid proteins. I was wondering whether there's any evidence, or if it could be conceivable that the males could somehow try to minimize the female resistance to the cost by somehow adapting the strategies they employ in a plastic way, and whether you see this at all in the sex ratio experimental evolution lines.
Yeah, that's a terrific question. Thank you. I think, I mean, I have this idea that, so there are multiple seminal fluid proteins, and I talked, you know, the latest estimate is over 200. And one question is, you know, why so many? Why is that so complex? And another feature of this mix is that there's a lot of redundancy in there. So there are multiple members of protease inhibitors. So multiple peptides and prohormones seem to do very similar things. So on the face of it, it looks kind of sort of too complex, if I put it that way. And so it's possible, and this is all just conjecture, but I have wondered and haven't yet got round to testing, but I think it would be good, including in the experimental evolution lines, which you talked about, whether as a whole, the strategy using a sort of multiple ways into influencing the female's behavior and physiology is partly to make it difficult, to render it difficult to evolve resistance. I mean, clearly they can, and I'm viewing this in the way that people think about strategies used in hospitals to prevent the evolution of resistance in antimicrobial resistance. And so there people talk about combinatorial or strategies or serial use of this, that and the other. And so it would be, you know, it's my hypothesis that is partly what is going on here, that some of that complexity is precisely to make it difficult to evolve resistance. And in a way, females will talk about the idea of a sensory a sensory bias. I mean, females need to respond to all of these molecules in order for reproduction to happen effectively. So they can't not respond at all, but they don't want to respond too much, and that makes it really females. So in terms of knowing whether any of that is part of, well, any of that is true at all, or any of that is true in terms of the what's happened in the experimental evolution lines, we don't yet know, but I think it would be really interesting to test, and I think there are ways to do it. So thanks.
Great, thank you. Very comprehensive answer. So I'll move on to one of the questions which is coming from our audience. So the question asks, your results show that males require multiple sensory cues for their social responses to other males, whereas females seem to only require touch for their responses. Do you know why you see these sex-related differences in sensory inputs?
No, but I can speculate, and I would be interested in thoughts on this, because the responses in females are very new. You know, this is something which we've just been working on. So the signaling theory, I think, is really interesting on the use of why individuals might need, you know, a single cue that has multiple components within it, or you have multiple different cues which individually signal different things. And so perhaps that's part of the reason. I mean, they're clearly not cueing into the same kinds of things. And so maybe another part of it is that perhaps the sexual and competitive environment from a male is more difficult to kind of pick up. So there are individuals sort of going in and out all the time, you know, moving around, and maybe that makes it a situation in which there's a lot more noise. And so males don't want to trigger and make this investment in making more ejaculate, and which we know is costly. You know, we know if males do this repeatedly, it impacts on their later life fertility. So you don't want to get that wrong. So you only want to do it when it's really reliable. So perhaps you need the complex sets of cues in order to only make that response so it's robust and reliable to a lot of behavioural noise. Whereas if the nature of the cues, you know, about the overposition environment is something which is kind of more long lasting, you know, a deposit is there, and it's going to be there for a while, it's not going to sort of trog off somewhere else, then maybe that's part of the reason. So I mean, I'm just sort of speculating, but I think these sorts of reasons might be part of the explanation.
Excellent, thank you very much. So we have another question which asks, do you know if the reduction in egg laying in response to exposure to other females is a reduction in resource investment or a change in provisioning, i.e. are the eggs they lay larger in anticipation of increased competition?
Yeah, I don't know. We don't know that. I mean, that's a really good question. Again, it's something that we could measure. So I think, you know, I'm writing all of this down, but I think that's a really good idea. I mean, it's possible that you could view it. Okay, I mean, the way I talked about it was a response of females to other females, and leading to a reduction in fecundity after mating, and maybe that's to avoid resource exploitation. It may be that on the flip side, the individual females that find themselves on their own, they increase or have higher fecundity, perhaps because they need the density dependent benefits of lots of larvae tunneling through the medium. And so, you know, there could be that sort of thing too. But I think the kind of provisioning and what's in there, both in terms of size, and something, you know, that we're really interested in is the way that mothers place deposit messenger RNAs and other molecules in the eggs that they lay, and whether that can vary according to the nutritional environment of the mothers, or even the social and sexual environment of the mothers. And I think that's an interesting one. So in response to the question, I think, yes, that's absolutely something we need to measure, but we don't just need to measure the size, you know, we need to look at what's in there as well. And I think we can do that. So thanks for the suggestion.
Brilliant, thank you. So we have a question which asks, do you know whether there is a minimum time frame of exposure to various social environments required for seeing the plastic responses you saw in males and females housed with the same sex individuals?
Yeah, again, great question, thank you. Yes, we do. In males, the males need around about 18 hours or so of exposure to other males in order to make these responses. I mean, we don't, and females need up to 48 hours, so it takes slightly longer for females to kind of cue in. In females, we probably need to do a few more sort of time points, but it looks like it's a little bit longer. So it does take some time for these responses to be initiated, and partly, I suppose, again, thinking about the plastic responses of this type, they are initiating potentially costly investment or significant investment decisions. So they need to be based upon reliable information. So that's not only coming from the cues, but it's from the perdurance of those cues, you know, how long they're experienced. I don't know why it would take longer for the responses to develop in females than in males. Perhaps that's to do with the sort of physiology involved. And work from Amanda Bretman's lab has also shown the details of the type of memory that's involved in males. And so males that have particular kinds of memory defects don't make these decisions very well. So they're really nice opportunities to kind of probe exactly what's going on there.
Thank you, that's really fascinating. It's nice to see the investigation from various different levels, both the behavioural, the transcriptomic, and also looking into the brains as well. That's fascinating. I think we'll just end with one final question. We're approaching the time. There's a question which is interested in the fitness consequences, and particularly wonders if the sensory plasticity in males and females is costly in terms of lifespan, and if that's something you've investigated.
Yeah, good question. I mean, and it's I would say it's important. It's really important to put all of this work into a proper fitness-based context. I mean, we have measured indices of fitness for many of the things that I've talked about, but clearly, you know, I mean, that's the currency. That's really what we need to make sure that we are focused on. So in males, in the male plasticity, what happens if you the survival of males that continually make longer matings, and so in response to conspecifics, that shortens their lifespan, and they have a marked drop-off in late life fertility. So it does appear that is costly. I mean, this is kind of interesting in the context as well of how people think about, you know, sperm being cheap and males not making much of an investment. Actually, the investment that they're putting in these seminal fluid proteins is quite significant. Anyway, so yes, in males. In females, we don't know yet. I mean, what we want to do, what we are trying to do, well, both in terms of how costly it is for females that continually make those responses, but also the benefits they get from the changes to fecundity patterns. Those are both things that require more work, I think.
Fascinating. Thank you very much. Some interesting directions to follow up in the future, for sure. Great. So I think that's all we have time for today, but it is our great pleasure to thank Tracey once again for a truly fascinating talk. Thank you very much.
Thank you. Thanks.
And for everyone else watching, next Wednesday we will be hosting Professor Anjali Goswami from the Natural History Museum in London, who will present on reconstructing the evolution of diversity with a phenomic approach. So we hope that you can join us then at the same time and same place. And in the meantime, check out our updates on Slack and our Twitter feed and spread the word about our seminars. Thanks for taking part, and until next time, stay safe and see you again soon. Goodbye.